Hello guys, good evening everyone. I welcome you all on Unacademy, India's largest learning platform. We are here at the channel that is Let's Crack NEET PG. Myself, Dr. Nidhi, I am alumni of King George Medical University, KGMU, Lucknow. I am teaching you all physiology, and guys, we all know physiology weightage is so much high. Every year 15 to 20 question in NEET PG examination come for sure. So everyone clear the concept of physiology so that you can score more in your exam and you will be able to achieve your target. You will be able to achieve your dream college, everyone. Good evening, Dr. Prakhar. Everyone download the Unacademy learning app from Play Store and follow me out there. My page is unacademy.com/@KGMC. Good evening, Dr. Shashank. इसमें एक प्लस कोर्स चल रहा है, वो भी आप ज्वाइन कर सकते हो by using my referral code that is Dr. Nidhi, and I have taken many more special classes which you can access anytime. There are many other renowned educators on this platform, so if you join that platform, you will be definitely benefited from these classes. Now coming to the subscription. First coming to the Plus subscription. In Plus, what are the benefit of Plus subscription? You will able to attend the daily live classes. In Unacademy, daily live classes are conducted, and in live classes you can interact with your educator and you can clear your doubt in live classes only. There are many more live tests and live quizzes are there. From that you can analyze your preparation. There are many batch courses, capsule courses are going out there. You can join that. There is a structured schedule out here. So if you follow that schedule, you will definitely be benefited and you will definitely able to achieve your target, everyone. Now coming to the Iconic subscription. In Iconic subscription, these plus benefit will be there, and along with that you can access the prep ladder. In prep ladder you can access the video lecture, you can access the question bank, you can access the rapid revision course, and you will be also provided with handwritten notes of prep ladder. So if you want that, you can go for the Iconic subscription. Now coming to the subscription fee. Iconic subscription starts from 1 year only. For 1 year the fee is 55,000. For 18 month the fee is 66,000. For 24 month fee is 77,000 and 36 month की fees है 92,000. After using my referral code, this will be more economical for all of you. You will get instant 10% discount in your subscription, and 12 month fee will reduce to 49,500. 18 month fee will reduce to 59,400. 24 month fee will reduce to 69,300, and 36 month fee will become 82,800. So that will be more economical for all of you. So do use my referral code that is Dr. Nidhi. Chalo. Now coming to the Plus subscription. Plus subscription are also available for 1 month, 3 months, 6 months. According to your requirement, you can take the subscription. But if you are planning to give the examination this year only, I will suggest go for 1 or 3 month subscription. And if you are planning to give the examination for the next year, I will highly suggest everyone to go for the 1 year, because you will get enough duration to revise your topic, to cover your topic. So go for the 1 year if you are planning to give the examination next year only. Final year students और third year students ज़्यादा टाइम का ले सकते हैं, 18 months, 24 months, 36 months, ताकि उनको ज़्यादा टाइम मिल जाए, ज़्यादा से ज़्यादा आप लेक्चर अटेंड कर सको और ज़्यादा से ज़्यादा बार रिवाइज कर सको. फ़ी की बात कर लेते हैं तो 1 month fee is 5,000, 3 month fee is 12,500, 6 month fee is 22,500, 12 month fee is 27,500, 18 month fee is 32,500, 24 month fee is 40,000, and 36 month fee is 50,000. After using my referral code, you will get instant 10% in discount, and your subscription fee will become more economical for all of you. 1 month fee will become 4,500. 3 month fee will become 11,250, 6 month fee will become 20,250, 12 month fee will become 24,750, 18 month fee will become 29,250, 24 month fee will become 36,000, and 36 month fee will become 45,000. So do use my referral code, everyone, that is Dr. Nidhi. Everyone follow me on Telegram. The page is NEET Physiology or Unacademy Physiology. Search this page and follow me out there so that you will get notification of every class and you won't miss a single class, everybody. Now coming to our topic. Today we are going to start muscle physiology. First we will study about the skeletal muscle. First is structure and function. Now first coming to the structural component. The cell membrane of a skeletal muscle is known as sarcolemma. Cell membrane of a muscle is known as sarcolemma. Now coming to the connective tissue layer. There are three connective tissue layers in our skeletal muscle: epimysium, पेरीमाइसियम, एंडोमाइसियम. epimysium क्या करता है? पूरी मसल की आउटर लेयरिंग है epimysium. What is perimysium? Perimysium is covering this fascicle. What is endomysium? It is the connective tissue layer which is covering a single muscle fiber. This single muscle fiber is covered by endomysium, and fascicle is covered by perimysium, and whole muscle is covered by outermost covering, that is epimysium. Clear? चलो, बहुत सारे muscle fibers join करते हैं and it form fascicle, and a group of fascicles join to form muscle. Clear? Single muscle fiber is covered by epimysium. ठीक है? और fascicle is covered by perimysium. Muscle is covered by epimysium. ठीक है? Clear? Muscle fiber is covered by endomysium. Fascicle is covered by perimysium. And muscle is covered by epimysium. Clear? Now coming to the functional unit of muscle. The functional unit of muscle is our sarcomere. ठीक है? अब आते हैं the most important part, that is structure of sarcomere. So what is sarcomere? Anyone? All the content between two Z line is our sarcomere. ठीक है? And this sarcomere contains certain bands and certain line. First coming to the band, it consists of A band, it consists of I band, it consists of H band. And it consists of two lines, Z line, M line. First coming to the A band. A band, it is also known as dark band. And it is made up of myosin, which is a thick filament, ठीक है? And it is seen in the centromere or center of sarcomere. Clear? A band is dark band, it is made up of myosin and it is present in the center of sarcomere. This is a diagram. See, this is thick band and this thick band is made up of myosin, and this thick band, this is our A band. Clear? What is A band? It is thick band containing myosin and it is present in the center of sarcomere. These two are Z line, and all the content between these Z line is our sarcomere. Clear? Now coming to the next band, which is I band. I band is also known as light band. It is made up of actin filament, which is thin filament. Clear? Now coming to the next band, H band. It is the portion of A band, यानी जो thick filament है उसका वो portion that is not overlapped by actin is known as H band. Clear? Now coming to the lines. M line. What is M line? It is the middle part of myosin. What is Z line? Z line is the line जिससे हमारा actin filament anchored होता है. ठीक है? See this diagram. This is A band containing thick filament. This is I band containing our thin filament, that is actin filament. And this non-overlapping part, let me change the color. This non-overlapping part of A band is known as H band. Clear? M line present at the center of sarcomere. Z line, it is the line where our actin filament is binded with the Z line. Clear? These terms are clear? Chalo. See this diagram. Here we are seeing some proteins that are important. First is alpha actinin. What is alpha actinin? It is the protein, anchoring protein, that is binding our actin filament to Z line. Clear? This is our alpha actinin. What is it doing? It is connecting our actin filament with the Z line. Clear? Next is titin. Next anchoring protein is titin. What is it? It is Z-like or a spring-like protein that is extending from Z line to M line. Clear? Next is M line proteins such as myomycin, M protein and obscurin. These three are M line protein. ठीक है, ये क्या करता है? This bind the myosin with M line. Clear? Beside that, there are some other anchoring protein, desmin, dystrophin. Clear? These are important anchoring protein. See, this is titin, जो हमने अभी diagram में देखा. They are the spring-like protein that is extending from Z line to M line. Next, जो हमने देखा alpha actinin. What is it doing? It is connecting I band with Z line. Clear? Next is myomycin. Myomycin is present in M line and it is connecting myomycin to M line. Clear? Now coming to the most important part, that is contraction of muscle. What happened to the bands when muscle contract? This is under relaxed state and this is under contracted state. ठीक है? In contracted state, we are seeing this H zone. This H band is decreasing. This I band, this is also decreasing. ठीक है? But the A band, what is A band? Here the A band is this one. And here the A band is also this one. So what happened? During contraction, our H band and I band shorten while A band remain unchanged. Clear? Everyone, clear? Sure. Now coming to the protein in our myofibrils. First, the most important is contractile protein. कौन से होते हैं? Our actin, our myosin. These are the major contractile protein present in our muscle. Beside that, there are regulating protein which are also present in our muscle. इसमें जो most important regulating protein है, that is our tropomyosin. This tropomyosin, what happened? Under resting condition, this tropomyosin cover the myosin binding site. This myosin binding site is present in actin and this tropomyosin bind to the myosin binding site in actin. So there will be no actin-myosin interaction. Clear? And during contraction, what happened? Contraction occurs in the presence of calcium. So this calcium goes and bind to the troponin C. And thus tropomyosin and troponin C complex move away. तो फिर हमारा myosin binding site क्या होगा? Expose हो जाएगा and our actin-myosin interaction will start. Clear? Everyone, resting condition में क्या होगा? Tropomyosin covers the myosin binding site, so there will be no actin-myosin interaction. और during contraction what happened? Calcium के presence में contraction होगा and this calcium bind to the troponin and tropomyosin troponin complex move away from the myosin binding site. Myosin binding site get exposed and actin-myosin interaction will start and our contraction will occur. Clear? This is the diagram. This is myosin binding site and during resting condition what happened? This tropomyosin covers the myosin binding site. During contraction what happened? It occurs in the presence of calcium. This calcium bind to the troponin C And what happened? This tropomyosin and troponin complex moves away from the myosin binding site. Myosin binding site get exposed and thus actin-myosin interaction occurs. Clear? Sure. Now coming to the next regulating protein, that is troponin. It consists of three parts: troponin I, troponin T, troponin C. Troponin T, T stand for tropomyosin. This troponin T binds with tropomyosin. This is troponin T, this binds with the tropomyosin. Now next, troponin I, that inhibit the myosin-actin interaction. This is troponin I, and this bind with actin and thus prevent the actin-myosin interaction. Next is troponin C. This is troponin C, and it has 4 calcium binding sites. Clear? The parts of troponin clear? Troponin I binds with tropomyosin, troponin T binds with tropomyosin, troponin I inhibit the actin-myosin interaction, troponin C have calcium binding sites. Clear? Now coming to the supporting proteins. First, we already studied titin, which is a spring-like protein connecting our Z line to M line. Next supporting protein is jasmine. This is the tumor marker of sarcoma. Remember this point, everyone. Next supporting protein is dystrophin. It undergo mutation, so that will cause muscular dystrophy. If this dystrophin will go mutation, it will cause muscular dystrophy. Clear? Now the next protein, that is relaxation protein. The most important one. Remember this one. Sarcoplasmic reticulum calcium ATPase pump. This is also known as Sarca pump. And this is very much important for relaxation of our muscle. So do remember this point. The relaxation protein is Sarca pump, that is sarcoplasmic reticulum calcium ATPase pump. Clear? Sure. Now coming to the most important part, how the excitation-contraction coupling occurs. ठीक है? Resting membrane potential of alpha motor neuron is minus 70 millivolt. What happened? Whenever this neuron is excited, what will happen? Depolarization will start, and during depolarization what happens? Sodium influx starts, and this sodium influx is via the voltage gated sodium channels. ठीक है? Due to this sodium influx, what will happen? RMP falls to minus 40 millivolt, and when the voltage becomes to minus 40 millivolt, calcium channels get open. These voltage gated calcium channels get open and calcium influx will start. What will happen then? This calcium will bind to the neurotransmitter vesicles. ठीक है? And there will be release of acetylcholine from neuromuscular junction. ठीक है? Clear? Resting membrane potential of alpha motor neuron is minus 70 millivolt. During excitation, what will happen? Depolarization start. Depolarization is due to the sodium influx, and this sodium influx is via voltage gated sodium channels. ठीक है? And हमारा RMP falls to minus 40 millivolt. जैसे ही minus 40 millivolt पे RMP आएगा, तो voltage gated calcium channels get open and calcium influx start होगा, और जो न्यूरॉन्स में हमारा न्यूरोट्रांसमीटर्स वेसिकल्स हैं वो से जाके बाइंड करेगा और हमारा वेसिकल रप्चर होगा और न्यूरोमस्कुलर जंक्शन पे एसिटाइलकोलिन रिलीज्ड हो जाएगा. क्लियर? चलो, दिस neurotransmitter is stored in the form of vesicle, which is known as quantus. Clear? What is quantus? It is the vesicle in which neurotransmitter is stored in alpha motor neuron. ठीक है? Now coming to the clinical part. Tetrodotox toxin. This toxin is present in puffer fish. What does it do? It inhibits the voltage gated sodium channel. So there will be no muscular contraction after having tetrodotox toxin. Clear? What happened in Lambert-Eaton myasthenia syndrome? This auto-antibodies is formed against the voltage-sensitive calcium channels. ठीक है, कैल्शियम चैनल्स के अगेंस्ट ऑटो एंटीबॉडी बनने लगेगा, कैल्शियम इन्फ्लक्स नहीं होगा. इट विल लीड टू माइस्थीनिया सिंड्रोम. दैट माइस्थीनिया सिंड्रोम इज लैम्बर्ट-इटन माइस्थीनिया सिंड्रोम. क्लियर? These two are important. Remember this point. Tetrodotoxin block the sodium channels, and Lambert-Eaton myasthenia syndromes autoantibodies are formed against these calcium channels. Clear? Everyone? चलो, अब क्या होगा? This acetylcholine will bind to the nicotinic acetylcholine receptor present in our skeletal muscles. That will lead to the sodium influx, and due to this sodium influx, what will happen? End plate potential या फिर local potential will be generated, and this local potential will transmitted along the sarcolemma. ठीक है? Clear? यहाँ तक clear है? Everyone, excitation contraction coupling तभी समझ में आएगा जब यहाँ तक सभी को क्लियर है. Everyone, चलो अब बात करते हैं एक्शन पोटेंशियल जो जनरेट हुआ, लोकल पोटेंशियल जो जनरेट हुआ, दैट विल रीच टू द सार्कोलेमा, एंड दिस सार्कोलेमा हैज अ प्रोजेक्शन विच इज नोन एज T-tubule. This is our sarcolemma. And this sarcolemma has a projection forming a T-tubules. So this action potential reaching the sarcolemma goes to the T-tubule. Clear? Now what happened? Dihydropyridine receptors are present in our T-tubule. This is important to remember. Dihydropyridine receptors. These are present in T-tubule. What will happen? Action potential reach the T-tubule and it will cause the conformational changes in this receptor. So what will happen? Our ryanodine receptors are present in the sarcoplasmic reticulums open. See, this is the diagram. Let me draw for all of you. This is sarcolemma. This is our T-tubule. This is our terminal cistern of, see, this is T-tubule. This is L-tubule. This is cistern of L-tubule. And L-tubule is formed by sarcoplasmic reticulum. ठीक है? This dihydropyridine receptor में conformational change होगा and it will open up the ryanodine receptor. This is ryanodine receptor. This is dihydropyridine receptor. ठीक है, this is sarcolemma. Action potential reach to the sarcolemma, it goes to the T-tubule, it causes conformational change in dihydropyridine receptor, and due to this ryanodine receptor channel open and हमारा calcium efflux will start. ठीक है? Clear? Everyone, in a skeletal muscle there is known as triad. What is triad? It consists of one T-tubule. Each sarcolemma contains one T-tubule and two L-tubules. ठीक है? So next tubule will be somewhat here. This is also L-tubule. So this is a classical triad we are getting in a skeletal muscle. Clear? Everyone, यहाँ तक clear है? चलो, हमारा ryanodine receptors will be open, and due to this, the release of calcium occurs from the terminal cistern of L-tubule. ठीक है? This calcium will go and bind to the troponin C. ठीक है? अब क्या होगा? This troponin and tropomyosin complex move away. This will move away and myosin binding site present in actin will be exposed. Clear? Further क्या होगा? जैसे ही expose होगा, actin myosin interaction होगा, and this will cause cross bridge formation, and there will be power stroke, and there will be our sliding of muscle, or you can say muscle contraction, sliding, or you can say muscle contraction. Clear? Up during relaxation, what happened? Calcium is sent back into the sarcoplasmic reticulum via channel, that is relaxation protein. अभी पढ़ा था हमने सरका pump, sarcoplasmic reticulum calcium ATPase pump. इस pump के through calcium is given back into the sarcoplasmic reticulum. ठीक है? और हम फाइनली क्या होगा? मसल रिलैक्सेशन. क्लियर? एवरीवन क्लियर है? कैसे क्लियर है सभी को? अल्फा मोटर न्यूरॉन से एसिटाइल कोलिन रिलीज होगा, एंड दिस एसिटाइल कोलिन विल बाइंड टू मस्कार्निक रिसेप्टर. ठीक है? and हमारा local potential generate होगा, and this local potential will reach to the sarcolemma, and from sarcolemma it will go to the T-tubule, and it will cause conformational change in dihydropyridine receptor. Due to that, what happened? Ryanodine receptor open. ठीक है? Calcium efflux will start, and this calcium will bind to troponin C. Troponin tropomyosin complex move away from the actin binding site. ठीक है? एक्टिन में जो मायोसिन बाइंडिंग साइट है उसे मूव आउट कर जाएगा, एक्टिन मायोसिन इंटरेक्शन होगा, देयर विल बी क्रॉस ब्रिज फॉर्मेशन, देयर विल बी पावर स्ट्रोक, देयर विल बी स्लाइडिंग ऑफ मसल्स. ठीक है? एंड ड्यूरिंग रिलैक्सेशन व्हाट हैपेंड? This calcium is pushed back into the sarcoplasmic reticulum via the Sarca pump, that is sarcoplasmic reticulum calcium ATPase pump. ठीक है? Calcium move out, हमारा वापस से troponin tropomyosin complex will bind to the myosin binding site present in actin, and हमारा muscle relaxation will be there. Clear? Everyone यहाँ तक समझ में आ गया? चलो. This is the same diagram. This is our T-tubule. This is our cistern of L-tubule. Action potential will reach to dihydropyridine receptor. Conformational change होगा. Ryanodine receptor will open and calcium efflux will start. And during relaxation, this Sarca pump push back the calcium inside the sarcoplasmic reticulum, and muscle relaxation will occur. Clear? Sure. Now coming to the sliding filament theory. Various steps are there. Step 1 is ATP binding. At this step, myosin head is at 45 degree. ठीक है? इसी स्टेज में ATP बाइंडेड होता है मायोसिन हेड से, एंड जैसे ही ATP बाइंडेड होता है मायोसिन हेड से, ATPase एक्टिवेट हो जाता है, एंड दिस ATPase कन्वर्ट द ATP इनटू ADP प्लस फॉस्फेट. ठीक है? एंड विद दिस हाइड्रोलाइसिस ऑफ ATP, नेक्स्ट स्टेज स्टार्ट, दैट इज ATP हाइड्रोलाइसिस. इट इज द रेस्टिंग स्टेज ऑफ मसल. ठीक है? मायोसिन हेड जैसे ही ATP हाइड्रोलाइसिस होगी, मायोसिन हेड टर्न्स टू 90 डिग्री. फ्रॉम 45 डिग्री ATP बाइंडिंग स्टेप में हमारा मायोसिन हेड इज 45 डिग्री एंड ATP हाइड्रोलाइसिस के स्टेज में हमारा मायोसिन हेड बिकम एट 90 डिग्री एंड जैसे ही 90 डिग्री पे आएगा देयर स्टार्ट्स क्रॉस पेज फॉर्मेशन. Actin in the presence of calcium, actin myosin interaction starts. ठीक है, everyone clear? And next step में there is release of phosphate from the myosin, and due to the release of phosphate, myosin head again bend to 45 degree, and this trigger the power store, and this pull the actin filaments towards itself and leading to the sliding and muscle contraction. Clear? Everyone clear? Next step में ADP is release and dissociation of ADP भी कह सकते हैं स्टेज को. इस स्टेज में क्या होता है, ADP फ्री मायोसिन कॉम्प्लेक्स. ठीक है, ADP फ्री मायोसिन कॉम्प्लेक्स हमेशा बाउंड होगा एक्टिन से until उसको नेक्स्ट ATP नहीं मिलेगा. जैसे ही ATP मिलेगा वैसे ही मायोसिन हेड टर्न्स फ्रॉम 90 to again 45 डिग्री. क्लियर? एवरीवन क्लियर? स्टेप 5 क्लियर है? किसी को कोई डाउट तो नहीं है अभी तक? चलो, इसी वजह से हमारा रिगर मोर्टिस देखने को मिलता है. आफ्टर डेथ हमारा मसल स्टिफनेस होता है. क्यों होता है? बिकॉज ATP इज नॉट देयर. तो ATP फ्री जो मायोसिन हेड है दैट विल बी रिमेन बॉन्ड टू द एक्टिन. That's why we get muscle stiffness after death. That is rigor mortis. Clear? Sure. This is the diagram. This diagram में we are seeing that ATP is binded to our myosin head and myosin head is at 45 degree. ठीक है? ATP hydrolysis will start and there will be formation of ADP and phosphate. जैसे ही हाइड्रोलिसिस होगा हमारा मायोसिन हेड टर्न्स टू 90 डिग्री. क्लियर? 90 डिग्री पे आते ही हमारा एक्टिन मायोसिन इंटरैक्शन स्टार्ट इन द प्रेजेंस ऑफ कैल्शियम. ठीक है, तो वीकली बाउंड स्टेट है, एक्टिन मायोसिन इंटरैक्शन है. फिर नेक्स्ट स्टेज में फॉस्फेट इज रिलीज्ड एंड ड्यू टू द रिलीज ऑफ फॉस्फेट अगेन द मायोसिन हेड comes from 90 to 45 degree, resulting to the power stroke. And due to this power stroke, what happened? There will be sliding of muscle. And after that, what will happen? ADP will be released, and this ADP free myosin head remain bound to the actin until it gets the next ATP. And यही स्टेज में हमें रिगर मोर्टिस मिलता है. Clear everybody? Everyone clear? Chalo. This is rigor mortis. Due to the lack of ATP, myosin head stuck into the actin filament. So there will be stiffening after death. This is known as rigor mortis. Now coming to the next topic, that is skeletal muscle contraction. The most important part is length tension relationship. For maximum tension in a muscle, there should be optimal overlap between actin and myosin filament and there should be room to slide. अभी समझ में आ जाएगा शायद room to slide. confusion अगर किसी को होगा तो अभी याद रखो, for maximum tension there should be optimal actin myosin overlap and there should be room to slide. Let me draw a diagram for all of you. This is point A, this is point B, this is point C, this is point D. This is length axis, this is tension axis. At point A, the sarcomere length is 1.65 micrometer. ठीक है, तो इस केस में क्या होता है, एक्टिन मायोसिन ओवरलैप तो ऑप्टिमल होता है, बट क्रॉस ब्रिज भी फॉर्म ऑप्टिमल होता है, बट देयर इज नो रूम टू स्लाइड. ठीक है, एक्टिन मायोसिन स्लाइड नहीं करेगा, सो देयर विल बी लेस टेंशन as compared to point B and C. Clear? See this diagram. This is point A, this is point B, this is point C, this is point D. At point A, the sarcomere length is 1.65 micrometer. तो इसमें क्या देख रहे हैं, actin myosin overlap is optimal, but there is no room to slide. So the tension developed out here will be less. Clear? And at point B and C, what is occurring? There is optimal actin myosin overlap. So there will be optimal actin myosin interaction. So there will be optimal tension, or you can say maximum tension. ठीक है? And at point B, the sarcomere length increases. ठीक है? और इस सिचुएशन में देयर इज नो एक्टिन मायोसिन ओवरलैप, सो द टेंशन डेवलप विल बी लेस. क्लियर एवरीवन? क्लियर? एट पॉइंट ए sarcomere length is 1.65 micrometer, and at point B and C the sarcomere length is 2 micrometer. This is the length at which the tension developed in muscle is maximum. Clear everyone? ठीक है, sarcomere length at point B and C is 2 micrometer. At this, my actin myosin interaction is optimal, so there is maximum tension. At point D, there is no actin myosin overlap, so the tension will be lowest. Clear? Everyone reply. Chalo. Optimal length. There are two terms here. Optimal length, which is also known as resting length. ठीक है? Length of a muscle at which tension is maximum is known as optimal length or resting length. ठीक है? और अभी जस्ट हमने देखा 2 micrometer sarcomere length पे maximum tension होता है. So this 2 micrometer length is our optimal length. Clear? Next terminology is equilibrium length. जब मसल को बोनी अटैचमेंट से डिटैच कर देंगे तो जब मसल की लेंथ होगी दैट इज इक्विलिब्रियम लेंथ. क्लियर? ऑप्टिमल लेंथ इज रेस्टिंग लेंथ. रिमेंबर दिस पॉइंट. एट दिस पॉइंट देयर इज मैक्सिमम टेंशन एंड इट इज अबाउट 2 माइक्रोमीटर. क्लियर? चलो अब बात करते हैं. टेंशन आर ऑफ 3 टाइप्स. एक्टिन मायोसिन इंटरैक्शन की वजह से जो टेंशन होता है दैट इज एक्टिव टेंशन. ड्यू टू स्ट्रेच ऑफ टाइटिन प्रोटीन द टेंशन डेवलप्ड इज नोन एज पैसिव टेंशन. सो दैट ए टोटल टेंशन विल बी इक्वल्स टू एक्टिव टेंशन प्लस पैसिव टेंशन. क्लियर? एक्टिव टेंशन एक्टिन मायोसिन इंटरैक्शन की वजह से होता है. पैसिव टेंशन इज ड्यू टू द स्ट्रेचिंग ऑफ टाइटिन प्रोटीन. क्लियर? चलो now coming to the energy for muscle contraction. First 3 seconds में जो energy मिलती है that is from immediate, so that is ATP जो भी body में store होगा that will be used during first 3 seconds of muscle contraction. ठीक है? After 3 seconds for 8 to 10 seconds up to 8 to 10 seconds ATP is formed from the creatinine phosphate. Clear? और उसके बाद शुरू होता है ग्लाइकोलाइसिस. वहाँ से ATP रीजनरेट होती है एंड आफ्टर दैट एरोबिक मेटाबॉलिज्म से ATP जनरेट होती है. क्लियर? इमीडिएट सोर्स कब तक सस्टेन करता है? फॉर 3 सेकेंड. आफ्टर दैट फॉर अप टू 8 to 10 सेकेंड क्रेटिनिन फॉस्फेट से ATP जनरेट होता है. आफ्टर दैट ग्लाइकोलाइसिस से ATP जनरेट होता है. आफ्टर दैट एरोबिक मेटाबॉलिज्म से ATP जनरेट होता है. क्लियर? चलो, now coming to the next topic, that is muscle fiber. There are various type of muscle fiber. They are divided into type 1 and type 2. Type 1 are small fibers. Type 2 are large fiber. This small fiber is also known as red fiber because they are red in color. So the complete name of a type 1 muscle fiber is low oxidative muscle fiber. ठीक है. Large one is divided into two A, type 2A, type 2B. Type 2A is red in color. Type 2B is white in color. Type 2A is also known as fast oxidative glycolytic. And type 2B is also known as fast glycolytic. Clear? This is type 1, which is red in color. They are small in type 2A. This is also red in color, but they are large in size. This is type 2B, which is large in size, but they are white in color. Clear? Now coming to the difference part. Type 1 is known as low oxidative type. Type 2A is known as fast oxidative glycolytic type. Type 2B is known as fast glycolytic. ठीक है? Type 1 is small. Type 2 is large. ठीक है? Type 1 is red in color. Type 2A is also red in color, while type 2B is white in color. Due to the absence of myoglobin, it is white in color. Clear? And due to the presence of myoglobin, they are red in color. ठीक है? Due to the presence of myoglobin, there will be oxygen storage. There will be oxygen storage, and there will be no oxygen storage. Clear? Due to the presence of oxygen, ATP formation will be via aerobic pathway. ठीक है? इस situation में भी ATP will be formed via the aerobic pathway, and in this situation in type 2B, the ATP will be formed via the anaerobic pathway. So in anaerobic pathway, what will happen? There will be production of this lactic acid, and due to this accumulation of lactic acid, there will be pain, and this pain is responsible for fatigue. So this type 2B is fatigue prone, and these two are fatigue resistant. Clear? Everyone, यहाँ तक clear है? Now coming to the twitch duration. What is twitch duration? When a single stimulus is given, muscle will undergo one contraction and one relaxation. The time taken to undergo one contraction and one relaxation is known as twitch duration. As these fibers are small in size, it will take long duration to undergo contraction and relaxation, so the twitch duration will be large. And in this situation, they are large fiber, so the twitch duration is small. Clear? Now coming to the tetanizing frequency. What is tetanizing frequency? Anyone? Tetanizing frequency is equals to 1 upon contraction period. As the twitch duration is large in this case, so contraction period will also be large in this case. So tetanizing frequency is low for the type 1 muscle fiber, and they are high for type 2 muscle fiber. ठीक है? Now coming to the function part. This type 1 muscle fiber is responsible for long and slow contraction, and this type 2 are responsible for fine and skilled movement. Clear? These all are important difference. Do remember these points, everyone. Type 1 muscle fiber is also known as low oxidative fiber. Type 2A muscle fiber is also known as fast oxidative glycolytic muscle fiber. Type 2B is known as fast glycolytic muscle fiber. This type 2B are white in color due to absence of myoglobin. That's why there will be no oxygen storage. That's why there will be ATP generation via anaerobic pathway. That why there will be production of lactic acid. So that will be fatigue prone. Clear, everyone? Shall we proceed? चलो अब बात करते हैं मोटर यूनिट की. Type 1 muscle fiber will form slow motor unit. Type 2A muscle fiber will form fast fatigue resistant motor unit, and type 2B fibers will form fast fatigable motor unit. ठीक है? अब बात करते हैं order of recruitment of muscle fiber during muscle contraction. Type 1 firstly recruit होता है, then comes type 2, then comes type 2B. During standing, our type 1 muscle fiber work. During walking, our type 2A muscle fiber will work. And during running, our type 2B muscle fiber will work. Clear? The order of recruitment of muscle fiber? Clear? Everyone? यहाँ तक सभी को clear है? डॉक्टर अननोन इन्विसिटस, सभी मसल कॉम्बिनेशन में प्रेजेंट होते हैं हमारे बॉडी में, किसी भी जगह एक इंडिविजुअल मसल प्रेजेंट नहीं है, ठीक है, दे आर ऑलवेज प्रेजेंट इन कॉम्बिनेशन. क्लियर? चलो, नाउ कमिंग टू द फेनोमेना ऑफ टिटनेस. टू अंडरस्टैंड द फेनोमेना ऑफ टिटनेस, first understand the muscle twitch. What is muscle twitch? When one stimulus is given, muscle undergo one contraction and one relaxation. This is known as single muscle twitch. ठीक है, अब आते हैं multiple stimulus में. तो when we give multiple stimulus, in incomplete tightness what happened? There will be series of contraction and series of relaxation. Clear? This is incomplete tetanus. Now in complete tetanus what happened? Due to the multiple stimulus, what will happen? There will be sustained contraction and there will be no relaxation. Clear? Incomplete tetanus में क्या होता है? Due to multiple stimulus, the muscle undergoes series of contraction and relaxation. While in complete tetanus, what happened? There will be sustained contraction and there will be no relaxation. ठीक है, और यहाँ पे जो stimulus की frequency होनी चाहिए, that should be of tetanizing frequency. The stimulus frequency should be of tetanizing frequency to get complete tetanus. Clear? Everyone, clear? चलो, this is the diagram. This is our single twitch. One stimulus is causing one contraction and one relaxation. ठीक है, next situation is we are starting our multiple stimulus. तो initially जैसे ही stimulus की frequency हम बढ़ाना शुरू करेंगे, तो what will happen? There will be summing of contraction. ठीक है, after that क्या होगा? There will be incomplete tetanus. Incomplete tetanus में we will get series of muscle contraction and muscle relaxation. Clear? And after that, when the frequency of stimulus is more than tetanizing frequency or equal to tetanizing frequency, what will happen? There will be sustained contraction. And this sustained contraction is known as complete tetanus. ठीक है, डिफरेंस समझ में आया? इनकम्प्लीट टिटनेस में एंड कंप्लीट टिटनेस में इनिशियली जैसे ही स्टिमुलस की फ्रिक्वेंसी बढ़ाना शुरू करते हैं, वी विल गेट समेशन. द स्ट्रेंथ ऑफ कॉन्ट्रैक्शन एंड स्ट्रेंथ ऑफ रिलैक्सेशन इंक्रीजेस. दिस विल कॉल एज समिंग ऑफ कॉन्ट्रैक्शन. आफ्टर दैट क्या होगा? इनकम्प्लीट टिटनेस होगा. वी विल गेट सीरीज ऑफ कॉन्ट्रैक्शन एंड सीरीज ऑफ मस्कुलर रिलैक्सेशन. After that, when the frequency of stimulus is more than tetanizing frequency or equal to tetanizing frequency, there will be sustained contraction and there will be no relaxation. This is known as complete tetanus. Clear? Chalo. Same formula, tetanizing frequency is 1 upon contraction period. ठीक है? अब next है the height of contraction. सिंगल सिंपल मसल ट्विच से कंपेयर करें तो कंप्लीट टेटनस में जो हाइट ऑफ कॉन्ट्रैक्शन है दैट इज 4 times ऑफ इनिशियल सिंगल ट्विच. ठीक है, डू रिमेंबर दिस पॉइंट. टेटनाइजिंग फ्रिक्वेंसी इज इक्वल टू 1 upon contraction period and contraction period should be in millisecond. Height of contraction in tetanized muscle is equals to 4 times of height of contraction in simple muscle twitch. Clear? Everyone? यहाँ तक clear है सभी को? चलो. Now coming to the question parts. First question. In muscle contraction, all are true except option A band remain unchanged. In muscle contraction, all are true except option A band remain unchanged. Option B, H zone disappear. Option C, I band become wider. Option D, 2 Z line come closure. Everyone, I am giving the timer. सभी लोग आंसर कीजिए. Everyone answer this question. In muscle contraction, all are true except. We already discussed A band remain unchanged. H band, H zone and I band क्या होता है? This is true. A band remain, sorry, this is false. A band हमारा remain unchanged. This is true. Sorry, this is true. H band disappears. H band disappear होता है. This is also true. I band, this also shorten up, not wider. So answer is C. Clear? Next question. The site where myosin head bind to actin in a skeletal muscle is covered by tropomyosin, troponin, calcium, none of the above. Everyone answer this question. The site where myosin head binds to actin in skeletal muscle is covered by. It is covered by tropomyosin. Answer is A. Next question. Question tropomyosin. Option A, help in fusion of actin and myosin. Option B, cover myosin and prevent attachment of actin and myosin. Option C, slides over myosin. Option D, causes calcium release. Everyone answer this question. True statement regarding tropomyosin. Answer is B. Tropomyosin cover the myosin and prevent the attachment of actin and myosin. Very good, everyone. Next question. Which of the following trigger muscle contraction? Option A, calcium binding to the tropomyosin. Option B, calcium binding to the troponin C. Option C, ATP breakdown. Option D, calcium binding to the troponin I. Which of the following trigger the muscle contraction? Everyone answer this question. Which of the following trigger muscle contraction? Calcium binding to the troponin C and ATP breakdown. Both are required for muscle contraction. So answer is both B and C. Twitch of a single motor unit is called as myoclonic jerk, fasciculation, tremor, chorea. Everyone answer this question. twitch of a single motor unit is called as? It is called as fasciculation. There is two terms, remember this point, fibrillation and fasciculation. Fasciculation is contraction of motor unit. Fibrillation is contraction of single muscle fiber. ठीक है? And this fasciculation can be seen through the skin, and this fibrillation cannot be seen through the skin. So do remember this point, fibrillation and fasciculation. Clear? Chalo, next question. The force of muscle contraction can be increased by all of the following except option A, increasing the frequency of activation of motor unit, option B, increasing the number of motor unit activated, option C, increasing the amplitude of action potential in motor unit, option D, recruiting large motor unit. Everyone answer this question. The force of muscle contraction can be increased by all of the following except option A, increasing the frequency of activation of motor unit. Firstly, हम in frequency add on करते हैं, तो it will cause summation. So this summation will increase the force of contraction. True. Increasing the number of motor unit activated. No more number of motor unit activated, more number of muscle fiber will be activated, and more fiber will be activated. So there will be more force of contraction. True. Option C, increasing the amplitude of action potential in alpha motor neuron. This cannot increase the force of contraction. So answer is C. Clear? Everyone? Chill. Now coming to the next question. Feature of a slow twitching muscle is or are? Option A, contain large number of amount of myoglobin. Option B, composed of small fiber. Option C, contain large amount of glycolytic enzyme. Option D, white in color. Option E, it has high ATPase activity. Everyone answer this question. Feature of slow twitching muscle is or are? Everyone answer this question. Feature of slow twitching muscle is, it is red in color, it is a small in size, ठीक है? तो smaller fiber, large amount of myoglobin, because the presence of myoglobin is there, so it is undergoing oxidative pathway. So it has oxidative enzyme, not glycolytic enzyme. It is not red in color. It has high ATPase activity. So answer is A, B and E. Clear? Now coming to the next question. Which of the following is not a sarcolemma protein? Sarcoglycan, dystrophin, dystroglycan, polycan. Everyone answer this question. Which of the following is not a sarcolemma protein? Our polycan is not a sarcolemma protein. Sarcoglycan, dystrophin, dystrofoglycan, all are the sarcolemma protein. Next question. काफ़ी अच्छा क्वेश्चन है. There is mutation of the gene coding the ryanodine receptors. It will cause malignant hypothermia. Which of the following statement best explain the increase in heat production in malignant hypothermia? Option A, increase muscle metabolism by excess of calcium. Option B, thermic effect of blood. Option C, increase sympathetic discharge. Option D, mitochondrial thermogenesis. AIMS में 2005 का क्वेश्चन है. Everyone answer this question. There is mutation of a gene coding for the ryanodine receptor in malignant hypothermia. Which of the following statement best explain the increased heat production in malignant hypothermia? Good evening, Dr. Vishal. Very good, doctor. Answer is A, increase muscle metabolism by excess of calcium. What happened? This ryanodine receptor, इससे क्या होता है? Calcium efflux होता है, and this calcium bind to our troponin C, and this tropomyosin complex move away from the myosin binding site, and what will happen? Actin myosin interaction will occur, and due to actin myosin interaction, what happened? हमारा मसल कॉन्ट्रैक्ट कर दी है. क्लियर? एंड व्हेन दिस राइनोडिन रिसेप्टर इज म्यूटेटेड, तो उस केस में क्या होता है कि दिस कैल्शियम इफ्लक्स स्टॉप नहीं होता है. देयर इज कंटिन्यूस इफ्लक्स ऑफ कैल्शियम, एंड ड्यू टू दिस कंटिन्यूस इफ्लक्स देयर इज कंटिन्यूस मसल कॉन्ट्रैक्शन, एंड ड्यू टू दिस कंटिन्यूस मसल कॉन्ट्रैक्शन देयर इज हाइपोथर्मिया. Very good, everyone. Answer is E. Next question. The protein marked A in the Z disc in the following diagram is this A. It is titin, nebulin, myosin, alpha-actinin. Everyone answer this question. The protein marked A in the Z line in the following diagram is. Everyone answer this question. This is our I band and this is Z line. I band is connected with Z line with the help of which anchoring protein? By alpha actinin. So answer is D. Myosin is present in M line. ठीक है, यहाँ पे मायोसिन फिलामेंट प्रेजेंट होता है. This A is our alpha actinin. चलो, next question. Graphs below is showing a simple muscle twitch of a frog gastrocnemic muscle. Calculate the tetanizing frequency of this muscle. ठीक है, this is the graph. You have to calculate the tetanizing frequency. Everyone answer this question. Excuse me. Tetanizing frequency is equals to 1 upon contraction period. ठीक है, and from the graph we can see the contraction period is 40 millisecond. So 1 upon 40 millisecond, it will come around 25 hertz. Answer is A. Clear? Next question. Detachment of a myosin head from actin is caused by option A, change in troponin C configuration. Option B, attachment of ATP to the head. Option B, binding of ATP and phosphate to the head. Option D, pumping of calcium into the sarcoplasmic reticulum. Detachment of myosin head from the actin is caused by. Everyone answer this question. Answer is both B and D. Pumping of calcium back into the sarcoplasmic reticulum and attachment of ATP to the head is responsible for the detachment of myosin head from the actin. Clear? So answer is both B and D. Now coming to the next question. All are related to negative leukotriophy except. Option A, excess calcium within the cell. Option B, defective calcium removal through the sodium calcium exchanger. Option C, improper functioning of Sarca pump. Option D, excess catecholamine. Everyone answer this question. All are related to negative leukotriophy except. Anyone remember what is leukotriophy? What does term leukotriophy mean? Anyone? All are related to negative leukotriophy except. Leukotriophy term is used for relaxation. Negative leukotriophy means delayed relaxation. Excess calcium within the cell will cause delayed relaxation. Defective calcium removal through the sodium calcium exchanger will cause delayed relaxation. Improper functioning of Sarca pump will also cause delayed relaxation. Excess catecholamine causes positive leukotriophy. Okay, answer is D. Now this is the terminology. Chronotrophy is related to heart rate. Inotrophy is related to myocardial contractility. Dermatology is related to cardiac conduction velocity. Bathmotrophy is related to cardiac excitability. Leukotriophy is related to relaxation. Autorhythmicity is related to the heart initiate its own impulse at rhythmic interval, that is known as autorhythmicity. So this is leukotriophy. Okay? Everyone remember these points? Now the next question. All of the following are true about excitation-contraction coupling except. Option A, acetylcholine is released at the nerve terminal. Option B, calcium is pumped back into the sarcoplasmic reticulum during relaxation. Option C, calcium is released from sarcoplasmic during contraction. Option D, calcium bind to the tropomyosin to initiate muscle contraction. एम्स नवंबर 2013 का क्वेश्चन है. Everyone answer this question. All are true about excitation-contraction coupling except. Everyone answer this question. Acetylcholine is released at nerve terminal. True. Calcium is pumped back into the sarcoplasmic reticulum during relaxation via the Sarca pump. True. Calcium is released from sarcoplasmic reticulum during contraction via ryanodine receptor. True. Calcium bind to troponin C, not tropomyosin. So answer is D. Clear? Everyone? Sure. Last question. Most common type of calcium channel of a skeletal muscle. T-type, L-type, R-type, N-type. Everyone answer this question. The most common type of calcium channels in our skeletal muscle is T-type, L-type, R-type, N-type. most common type of calcium channel of the skeletal muscle is long lasting type? Answer is B. No, Dr. Anurag, answer is B. Long lasting calcium channel जो skeletal muscle में ryanodine receptor channels होते हैं, they are long lasting type. So answer is B. Clear? That's all for today. Thank you so much everyone for joining. Hope you like the session. If you like the session, do like and share with your friends so that many more can join. इसमें यार कंजूसी मत किया करो आप लोग. Do like and subscribe our channel, that is Let's Crack NEET PG, and press the bell icon so that you will get notification of every class and you won't miss a single class. मुझे फॉलो कर सकते हैं एवरीवन टेलीग्राम पे. टेलीग्राम पेज इज नीट फिजियोलॉजी और अनअकैडमी फिजियोलॉजी. सर्च कर लीजिएगा and you will get my page and follow me out there so that you will get notification of every class and you won't miss a single class. If you take the subscription, do use my referral code, that is Dr. Nidhi, to get instant 10% discount in your subscription. Learning from home can be difficult, but you can definitely achieve your target. You can definitely be in your dream college. All you have to do. Yes, Dr. Anurag, कल का session भी हमारा 5 PM होगा, and tomorrow we will study about cardiac muscle and smooth muscle. ठीक है? Everyone, do join tomorrow class also. ठीक है? Learning from home can be difficult, but you can definitely achieve your target. All you have to do is to go through your syllabus thoroughly, and after doing that, you have to do more and more MCQ. ठीक है? You will definitely able to achieve your target. Thank you so much everyone. We will definitely crack our exam with good rank. Do use my referral code if you take the subscription, that is Dr. Nidhi. Don't forget this referral code, that is Dr. Nidhi. Thank you so much everyone for joining. Most welcome Dr. Apur, most welcome Dr. Anurag.